Hello everyone, we the students of Indira Institute of Pharmacy, Sadavli Devrukh presenting the poster for ICEDS 2022 conference. We are glad for the acceptance of the poster. So the topic of our poster is assessment of microbial bioburden on used mask. As we all know, COVID-19 pandemic has distorted the workings of the entire world, and although there is no valid cure for coronavirus yet, there are various ways you can prevent yourself as well as others from getting an infection. Using a mask is one of the most effective ways to avoid COVID-19 infections. In the past months, there has been a surge in the use of masks. However, what's more important is the correct usage of masks and not just the usage. Primarily, there are three types of masks used: cloth mask, surgical mask, and N95 respirator mask. No matter which mask you use, it is important to use, reuse, and dispose of the mask appropriately and responsibly. Coming to our title, basically bioburden is defined as the number of microorganisms living on a surface that has not been sterilized. The aim of bioburden testing is to measure the total number of viable microorganisms prior to sterilization. So, this study was carried out for quantification of microbes on masks used by individuals during COVID-19 pandemic and to estimate the microbial population. Coming to methods of our project, first of all, we had prepared the media. The media used for quantification of microorganisms was de-engaged media. Total 100 masks were distributed between faculty members and students and informed them to use the mask for 24 hours and then collected the mask the next day in a Ziploc bag. Then the surgical masks were tested by immediately making an impression of the internal surface of the mask on sterile culture media and incubated for 24 hours in an aerobic humid atmosphere at 37 degrees Celsius. The growth of microorganisms was observed and the colony forming units were then counted on microbial bacterial colony counter. In this study, all the 100 samples showed the presence of the microbes. The graph represents that 33% masks are found to be heavily contaminated with colonies more than 300 CFU and are considered as TNTC, that is, too numerous to count. 15% masks found to be having colonies in between range 1 to 50. 17% comes in range of 50 to 100. 15% masks found in the range of 100 to 150. 9% found to have colonies in the range of 150 to 200. 4% found to have colonies in the range of 250 CFU to 300 CFU respectively. Therefore, based on the results of the present study, we conclude that surgical masks are a potential source of bacterial shedding and ideally it is suggested to avoid reusing the mask after using them once and should be used properly in proper manner. That is, it should be always removed using its string, making sure that you don't touch the front of the mask. Fit the mask snugly around the nose and must be completely covering your nose and mouth and not just hanging loosely around the neck. Otherwise, the purpose of using the mask is defeated. And while discarding, wrap the mask in newspaper, put it in a polythene bag and discard it. Finally, I would like to end by saying that you should consider the mask as a part of our professional life and try to convince people that there is no way of considering the mask as an enemy, but consider it as a protection, like an umbrella against the rain, like a coat against the cold, and as a sign of civilization to protect our colleges and people around us. Thank you.